So, I thought I'd make this video. I'm not sure if it's necessary since, we're meeting in class. But I'm teaching a section of the lab online, so this is for people that are taking the microbiology course online, or lab online. So I'll update this start here to watch this video. That'll change once I upload it and put it into this. I'll put the link here. It'll be a YouTube video. And I just kind of want to quickly go through what to expect. Being that it's online, there'll be videos to watch each week. Now, like for instance, some of the videos I have made, not for an online lab, but these were videos that I used to show my face-to-face labs, right? When we did the microscope, you'd watch this microscope or inoculating a streak play. I'd show that actually in the during the lab, So now I'm using these for the online lab. So they might appear a little bit different. Now I've got a bunch of these videos that are numbered, lab video 1, 2, 3, 4, 5, 6, and 7. Now these I made for the online course. So instead of actually doing the experiments that we do in one lab, this will be for like 1 week, this will be for another week, and the next week I'll say watch lab video for this week. That'll be your assignment. And it'll explain what we would have done in that lab, how the media work, and I'll kind of use the PowerPoint like I would in class and during the lab to explain the concepts that you expect to know for the quizzes and for the lab practical. Right now you can't see these videos. The little thing over here means that they're hidden from you for right now. But I'll open them up when we get closer to the time to watch them. So this is the, those are all found in the content section, like everything, that you really, like the lab PowerPoint's in the content section as well. So this is my information. When we used the lab manual, right, we used this one, so I put that there. It's not, you don't need to buy it. It's not necessary. I'm pretty sure Emily's not using it either. You will need online access, of course. If you have issues online, be sure to contact the help desk. I can't troubleshoot your computer or computer issues for you. This is completely online, and then instead of doing the labs themselves in the lab, you'll be watching lab videos. That's, what I expect you to do each week. So for instance, when you go to next week, there'll be an announcement for week 1. So I'll update these here. Week 1. This is the announcement you'll see next week. You should watch the culturing bacteria video, inoculating media, and streak plate videos in the content section. That's all you got to do. Watch those 2 videos. I might take a few notes while I'm watching them, right? Because you're expected to know the material on the PowerPoint as well as the material in these videos. Okay, so then week 2, you'll be assigned to watch parts of the microscope and use of a microscope. Take notes for the quiz. Quiz 1, and I'll have the correct date for that. This was for in the spring. So that's how you know what to do each week for the lab. Okay. So now there are what are called purpose and principles that are more or less homework assignments. I use these when we did the lab face-to-face too. So they'll be due there'll be P&Ps due not this Sunday, and I don't think there'll be any due the following Sunday, but at some point, you'll have due dates for P&Ps. They need to be completed. And in the announcement, when I announce it, I'll link it to where they're found. But in the quiz section, that's where you find the P&Ps and the quizzes. The P&P for the negative stain is not a quiz. There's a box that you put your purpose in. There's a box that you write in your principle. Right, that's what the P&P stand for, purpose and principle for the negative stain. Those are homework assignments, each P&P. The quizzes are like regular multiple choice quizzes that you've taken before. So for instance, the negative stain quiz is due September 12th, that's a Sunday, by midnight. So is the gram stain. The following week, you'll have to have these two P&Ps on September 19th. These two P&Ps are due, the purpose and the principle. Now, there are videos and instructions on how to complete the P&P assignments. So if you go to the content section, here's a Word file with instructions for the P&P. There's also, just want to make this clear, in the video, there's a video that you can watch as well. Right? There's a P&P stain instructions video. There's also a P&P biochemical test instruction video. So there's 5 stains that we do. You follow these instructions to do P&Ps or the stains. When we're done with those are the first, I already showed you that when they were due, right? Those 2 sets are due, and then all the rest of the remaining P&Ps will be for biochemical tests, right? So the biochemical tests are what we'll be doing in the remaining videos. So you can watch the video to help you understand what the principle of the experiment is, or most of the information you need to put in your purpose and principle are found on the PowerPoint. So if we go to that, let's see, do I have it on here? This is the lab PowerPoint. It even has instructions for doing P&Ps on that as well. These are like a quick snippet of how to do the P&Ps. It's on the PowerPoint. When we went online, I made the videos and other things. But if you take one, like for instance, the spore stain. What is the principle, or the purpose of the spore stain? It's right here, to identify spore-producing bacteria. That's what I expect you to put for when you open the P&P to do the P&P for the spore stain. It'll say, what is the purpose of the spore stain? This is the correct answer. What is the principle of the spore stain? Will be the next question, right? You'll get those 2 questions for each P&P. The principle would be the counter stain is malachite green. The counter stain is safranin. When you're done, the spore appears green. The non-spores appear red. Here's an example of what they would look like. You need to heat the flame, right? You need to heat up the primary stain on the slide. That's part of the procedure, right? So that should go in your principle as well. Okay? So all the purposes for each one that you have to do are written. Purpose. There's a purpose here. The top line is the purpose. The information you need to include in your principle is the remainder part of this, what you see on the slide. Now I want you to use complete sentences. So if you copy and paste, you might not get all 4 points because you didn't use complete sentences, right? You'd say the inhibitor of the eosinmethylene blue is eosinmethylene blue auger, right? The PMB auger indicator is eosinmethylene blue. Okay. Lactose is found in the media If it's fermented, it's indicated by a green metallic sheen, or a fishite, or Enterobacter. Okay, so if you read the instructions and if you watch the video and then you still have any questions, ask me in class about it. All right? I would prefer to talk to you about these or during office hours than, you know, to email me or not. I don't know what to do for the P&P. Please don't email me that. Ask me in person, please. Okay, so that's the most complicated part. It's easy to get all the points for your P&Ps. If you miss them, you know, you'll be able to make up a couple at the end or redo a couple, but you can't do both, right? It's one or the other. So don't miss the deadlines. There's 4 quizzes. Talked about that. There's a stain quiz. I'll talk more about that. I think there's even a video on the stain quiz. The unknown, it'll be covered in lab video number 7, what you have to do for that, or I might even talk about it in class. The practical, there's 20 multiple choice questions worth 1 point, and there's 40 questions that are more or less fill in the blank, and they're worth 2 points. And then this is the breakdown. You can earn a total of 340 points in lab. They get put into the grade book for your lecture. I add up both. That determines your grade for both lecture and lab. Right? There's not separate grades. And then the remaining part of this, right, is the schedule. Right? These are the videos you'd watch for week 2. Okay, quiz 1. I think quiz 1's not due until September 12th, right? There'll be an announcement about completing quiz 1. I should probably put quiz 1 down here. Quiz 2, you know, it'll cover labs. So this tells you what quiz 2 covers too. Labs 3 and 4, right? This lab and this lab are what you need to study for quiz 2. Labs 1 and 2 are the material you need to know for quiz 1. Quiz 3, labs 5, 6, and 7 are on quiz 3. So this is helpful for that sort of reason, right? You know, and then these are the lab videos. This week you'd watch lab video 1, then lab video 2, 5. There's a total of 7 numbered videos. And then there are these videos, like I say, these are videos that I made. They're not YouTube videos, right? I think they're found on some Daytona State server. I can't download them. No one can, right? You can watch it if you have the link. That's the only way. So anyway, I think that about covers it for the lab. I just wanted to give you you weren't quite clear when I talked about this in class. You know, this is to hopefully this is helpful for that if you were confused. So I'll see you later. Take care.